All right, welcome everybody. Today we're going to talk a little bit about DNA replication. All right, so just to review, before we talked a little bit about the cell cycle, and we talked about mitosis and meiosis and different types of cell division. But essentially, what we said is that the cell is going to be in what we call the gap 1 phase, the synthesis phase, and the gap 2 phase most of the time. And that's what we refer to as interphase, right? So this long period of time when the cell is not dividing is referred to as interphase. And then this short period of time when the cell does divide, okay, that's either going to be mitosis or possibly meiosis, depending on the type of cells that we're talking about. So today we're really going to focus in on this S phase where DNA gets replicated, and we want to go ahead and see exactly what's going on. So in the past, scientists thought, well, how is DNA replicated? They knew that DNA was double-stranded, but they weren't quite sure how it replicated. So two different ideas were floating around. One was that it was a semi-conservative. So if you think about the word semi is half, like a semicircle. Conservative means to save. So semi-conservative, half saved. Okay, and that was one idea, is that it was semi-conservative. And if we look here, we have a parent DNA molecule, so an original molecule, and then it copies itself, and we end up with two molecules that's half old and half new. Versus the conservative model, where we end up with one of the new strands that is completely the same as the parent, and then one totally new strand. So it turns out that the conservative model was wrong, and that DNA actually does replicate using the semi-conservative model. Okay, and with the semi-conservative model, each new double helix keeps one strand of the original DNA, and then the other strand is new. And in fact, the original strand is going to be used as a template. Okay? Or another way to think about that is that the information on the old strand is being used to make the new strand. So we say that the new strand is not going to be identical because it's not, but instead it's complementary. It matches up using the base pairing rule. So if there's an A on the old strand, there'll be a T on the new strand. If there's a C on the old strand, there'll be a G on the new strand. All right. So we have to think back to when we learned about the structure of DNA, and we learned about the structure of a nucleotide. So right here, this is showing a nucleotide, right? We have our sugar, which is their deoxyribose sugar, attached to a phosphate group, and the sugar is also attached to a nitrogen base. One of the things that we didn't get into when we talked about the sugar is that the sugar molecule, the carbons within the sugar, are numbered. So the sugar, or the carbon that's closest to the nitrogen base, is numbered number 1, and then it just goes around the sugar molecule. Here's carbon number 2, carbon number 3, carbon number 4, and carbon number 5, which attaches to the phosphate group. So all of the carbons get numbered. So this is really important to understand, because if we look at DNA, while it's double-stranded, the two strands are not parallel to each other. Yes, the two strands are running side by side, but they're running in the opposite direction. So we say that they are anti-parallel to each other. Okay? And so if we look at this, on this end, we would call it the 5' end because we have a 5' carbon at the top here. And we say that this is the 3' end because if you look here, we have a 3' carbon on this side. So if we go around 1, 2, 3, there's the 3' carbon right there. So on this strand of DNA, the 5' carbon's at the top, the 3' carbon's at the bottom. On the other strand, okay, the 5' is at the bottom and the 3' is at the top. Another thing you can do is you can look at where the phosphate groups. On this end, the phosphate group is up. On this strand, the phosphate group is down. So we say that the DNA is anti-parallel, the strands are to each other because they're running side by side, but they're going in the opposite direction. So that's really important to understand that because when we think about how we're going to build a new DNA molecule, it's also going to have to be anti-parallel. So pretty much the first step in this process of DNA replication is an enzyme. Okay, the name of the enzyme is called helicase. Remember that whenever you see the ASE at the end, okay, whenever we see that, ASE, okay, this means that it is an enzyme. Okay, and so helicase, okay, its job is to separate the two strands of the DNA, and that way the other enzymes will have access and be able to get to the nitrogen bases. Okay, so the helicase separates the two strands. Okay, the spot where the helicase separates the two strands is called the replication fork. And no, we're not talking about this type of fork right here. No, we're talking about this type of fork, okay, right here. Okay, like a fork in the road. Okay, and so here's our helicase. Okay, and it's separating the two strands, creating this replication fork. Okay, and the helicase is moving, right? So it's unzipping the DNA. We can think about it that way. Okay, as the helicase continues to move down the DNA molecule, the replication fork, of course, moves with it. All right, so this is where it gets a little bit tricky because remember we said the DNA is anti-parallel. So an enzyme called DNA polymerase III builds new DNA strands in the direction of 5 to 3. So we see here the DNA polymerase III is building in the 5 to 3 direction. Okay? It's doing this on something called the leading strand. So the DNA polymerase III moves towards the fork always on the leading strand. Okay, so when the DNA polymerase moves toward it, a new strand can be built continuously. So as it moves in this direction, okay, it's going to continue to unzip, and it just continuously builds a new leading strand. Okay, and just another look. Okay, this is showing the same thing, but in this picture, okay, we see that the leading strand is on top, and we're still building in the 5. Okay, so let me highlight that. Okay, we're building in the 5 to 3. I'll just write it in right here because they didn't put it. Okay, 5 to 3 direction. All right. All right. Now, if we look at what we call the lagging strand, okay, that would be the other strand of DNA, we see that, let me just go, oops, got a little ahead of myself. Okay, on the lagging strand, okay, the DNA polymerase can only build the new strand, remember, in the 5 to 3 direction. So that's true on both the leading and the lagging strand. But because the DNA is anti-parallel, that means that on the other strand, the DNA polymerase has to build away from the replication fork, still in the 5 to 3 direction. So if I write it in here, okay, we're moving in the 5 to 3 direction. We're building it, okay? So on the leading strand that's up here in the top, we build from the 5 to 3 direction. And on the lagging strand, we also build in the 5 to 3 direction. You probably noticed that on the lagging strand, there's more than one strand, right? There's multiple strands, right? And then we call them fragments. In fact, we call them Okazaki fragments. So these short little fragments are called Okazaki fragments, named after the scientists that discovered them. Okay? And eventually you can't have gaps because this one will run into this one. And so you can't have gaps. So eventually an enzyme called ligase is going to come in and it's going to connect the Okazaki fragments together, right? Bonding the sugar and the phosphate together and connects them. All right. All right, so if we look at how this process starts, it actually begins with two helicase enzymes. Okay, and they begin at the same point in the center, and they move away from each other. Okay, and since they're moving away from each other, okay, we have two replication forks. Okay, we have a replication fork right here and a replication fork right here. Okay, and this entire thing is referred to as the replication bubble. So as the helicase moves away from each other, the replication bubble is going to get bigger and bigger. Okay. All right. And it's also important to understand, you might be wondering, well, which is the leading strand and the lagging strand? So if we look at this top strand, okay, this top strand is going to have a leading and a lagging strand on it. So if this over here is the leading strand, so let me go ahead and just write this in, okay, if this is my leading strand, okay, going in this direction right here, okay, that would make this 5 and this 3. So that would mean over here, this would be the lagging strand because it would still be moving in the 5 to 3 direction. So this would be 5, okay, and this would be 3. Okay, so if this is the leading strand, then there's a lagging strand over here. And that would make this a leading strand down here and a lagging strand over here. So on each strand of the DNA, there's going to be leading and lagging strands. Let's look at it. Let's look at this again more closely. Okay, so like I said, pick my pen color real quick. All right, this is my leading strand. We're building from 5 to 3. Okay, and so on this side of the bubble, okay, we're still building from 5 to 3, But on this side of the bubble, we're building away from the replication fork, okay, because this is the lagging strand. And on this side, we're building towards the replication fork, because this is the leading strand. And on the other strand of DNA down here at the bottom, okay, here's our leading strand. We're building from 5 to 3, so we're building in this direction. Okay, but over here, we're also building in this direction from 5 to 3. Okay, but this is the lagging strand because we're building away from the replication fork. So if we look at the bottom, okay, we're building to the right. If we look at the top, we're building to the left. And again, that's because DNA is anti-parallel. So we always build in the 5 to 3 direction, which are opposite directions if we're talking about anti-parallel DNA. Okay, so this is just showing our DNA polymerase 3 and the direction that it moves in. Okay, as well, since we've talked about the replication bubble, okay, it turns out that the replication bubble it's not just one bubble, okay, but there are many bubbles forming, okay, and so there are multiple replication bubbles happening at once. Okay, this is kind of showing just a diagram of what that looks like. This is an actual picture of the DNA, so we can see. Okay, I'll draw it in here. Okay, there's a bubble. Okay, right here. There's a bubble right here, and there's a bubble right here. Okay, and so there's multiple bubbles happening. Eventually the bubble expand until they're going to expand until they reach each other and they merge together. Okay, this speeds up the process of DNA replication. So what I'd like for you guys to do right now is go ahead and get out a piece of binder paper. Okay, you can pause the video to do that. And then after you have your binder paper, I would like you to answer the question. Okay, or it's not really a question, but what I like you to do is write out, we talked about 3 different enzymes, okay, today in this PowerPoint. And go ahead and write out what those 3 enzymes are and what the job of each one of those enzymes is. Okay, and then I would like you to tell me what direction DNA is built in. Okay, and what is the difference between the leading and the lagging strand. Right, so you can go ahead and pause the video right now, answer those questions. If you need to scroll back and look at the video again, that's fine, but go ahead and pause it now and you can answer those questions. All right, welcome back, and that's all I got for you today. See you guys later.